14.2 Introduction. The use of natural enemies for the control of harmful insects, other living creatures like animals and plants is known as biological control. Basically, living organisms are used to suppress pest populations so that they are less damaging. Term biological control was first used by Smith in 1919. It is also called as biocontrol. In insects it relies on predation and parasitism or other natural mechanism. It is important for integrated pest management, IPM. Biological control agents includes predators, parasitoids, pathogens and competitors. There are three basic strategies for biological pest controls are 1. Classical importation. In this a natural enemy of a pest is introduced in the hope of achieving control. 2. Inductive augmentation, in which a large population of natural enemies are administered for quick pest control. 3. Inoculative conservation, in which measures are taken to maintain natural enemies through regular reestablishment. 319. Disadvantages of biological control are: it can have side effects on biodiversity through attack on non-target species when species is introduced without thorough understanding of its consequences on biodiversity. It can also disturb natural food chain. It is slow process. It is costly to start with. Lots of planning and money is required to start it. Advantages of biological control are: due to negative use of chemical pesticide on the environment and human health, there is growing need of utilizing biopesticides. No damage to the mammals and environment. Quick action and breakdown. Low phytotoxicity. Only targeted pests will be eliminated, no problem of resistance. 14.3 Parasites. The organisms that lives and feeds on host are called parasites. Parasite obtains food and shelter from host's expense. Most of the species used in biological control kills the host and are called as 320. Parasitoids. Many insects and microbial parasites are important biological control agents. Most of parasitic insects are either flies, order Diptera, or wasps, order Hymenoptera. Examples: adult females of parasites like wasps that attack scales and whiteflies feeds and kill their host. Thus they are important biological control and causes host mortality due to parasitism. Waterborne mold Legenidium giganteum parasitizes the larval stage of mosquitoes. They infect mosquito larvae as motile spores in water. This mold is resistant to desiccation and can remain dormant for many years. The fungus Ampelomyces quisqualis parasitizes powdery mildew fungi on several fruit crops. Likewise Bovaria bassiana fungus parasitizes many insects. Trichoderma harzianum fungus parasitize Pythium, Rhizoctonia and other pathogens. Trichoderma species parasitize soil-borne pathogens such as Rhizoctonia and Pythium. Even more interesting are soil-inhabiting fungi that trap and devour nematodes with specialized structures that resemble lollipops and lassos. Some bacteria are also parasitize nematodes. For example, the bacterium Pasteurella penetrans attacks the root knot nematode. Aphelinid wasps Aphidiinae, subfamily Braconidae, attack aphids. Trichogrammididae parasitize insect eggs. Aphelinidae, Encyrtidae, Euliphidae, and Ichneumonidae parasitize insect pests. Some more examples of parasites and their hosts are tachnid flies, caterpillars, beetles. Trichogramma wasps, moth eggs. Bacillus thuringiensis bacterium, butterfly, moth larva. Pseudomonas fluorescens bacterium, fungi. Polyhedrosis virus, butterfly, moth larva. Arthrobitrus nematode trapping fungus, nematodes. 321. 322. Steinerma nematode, insect larva. 14.4 Parasitiods. Parasitoids lay their eggs inside body of insect host. They are insects with an immature stage, mostly larva, that develops on or in a single insect host, and ultimately kills the host. They used host cells' resources for food of developing larva. Firstly, larvae grow on fatty tissues that allows host to grow normally. As it develops further, it utilizes host cells' vital organs and kill it. They are mostly patristic in immature stage but free living in adult stage. Parasitoids are mostly small flies or wasps. Few parasitoids attack one or two species and called specialists, while other attack large number of species, therefore are generalists. On the basis of mode of development, OF parasitoids can be categorized as endoparasitoid, internal, or ectoparasitoid, external. The adults are typically free living, and may be predators. They may also feed on other resources, such as honeydew, plant nectar or pollen. Because parasitoids must be adapted to the life cycle, physiology and defenses of their hosts, they are limited in their host range, and many are highly specialized. Thus, accurate identification of the host and parasitoid species is critically important in using parasitoids for biological control. They includes mostly wasps or flies. They have narrow host range. Commercially, there are two types of rearing systems for parasitoids. Short-term daily output with high production of parasitoids per day. These facilities are used. Parasitoid. Disadvantages of biological control are: it can have side effects on biodiversity through attack on non-target species when species is introduced without thorough understanding of its consequences on biodiversity. It can also disturb natural food chain. It is slow process. It is costly to start with. Lots of planning and money is required to start it. Advantages of biological control are: due to negative use of chemical pesticide on the environment and human health, there is growing need of utilizing biopesticides. No damage to the mammals and environment. Quick action and breakdown. Low phytotoxicity. Only targeted pests will be eliminated, no problem of resistance. 14.3 Parasites. The organisms that lives and feeds on host are called parasites. Parasite obtains food and shelter from host's expense. Most of the species used in biological control kills the host and are called as 320. Parasitoids. Many insects and microbial parasites are important biological control agents. Most of parasitic insects are either flies, order Diptera, or wasps, order Hymenoptera. Examples. Adult females of parasites like wasps that attack scales and whiteflies feeds and kill their host. Thus they are important biological control and causes host mortality due to parasitism. Waterborne mold Legenidium giganteum parasitizes the larval stage of mosquitoes. They infect mosquito larvae as motile spores in water. This mold is resistant to desiccation and can remain dormant for many years. The fungus Ampelomyces quisqualis parasitizes powdery mildew fungi on several fruit crops. Likewise Bovaria bassiana fungus parasitizes many insects. Trichoderma harzianum fungus parasitize Pythium, Rhizoctonia and other pathogens. Trichoderma species parasitize soil-borne pathogens such as Rhizoctonia and Pythium. Even more interesting are soil-inhabiting fungi that trap and devour nematodes with specialized structures that resemble lollipops and lassos. Some bacteria are also parasitize nematodes. For example, the bacterium Pasteurella penetrans attacks the root knot nematode. Aphelinid wasps Aphidiinae, subfamily Braconidae, attack aphids. Trichogrammididae parasitize insect eggs. Aphilinidae, Insyrtidae, Euliphidae, and Ichneumonidae parasitize insect pests. Some more examples of parasites and their hosts are tachnid flies, caterpillars, beetles. Trichogramma wasps, moth eggs. Bacillus thuringiensis bacterium, butterfly, moth larva. Pseudomonas fluorescens bacterium, fungi. Polyhedrosis virus, butterfly, moth larva. Arthrobitrus nematode trapping fungus, nematodes. 321. 322. Steinerma nematode, insect larva. 14.4 Parasitiods. Parasitoids lay their eggs inside body of insect host. They are insects with an immature stage, mostly larva, that develops on or in a single insect host, and ultimately kills the host. They use host cells' resources for food of developing larvae. Firstly, larvae grow on fatty tissues that allows host to grow normally. As it develops further, it utilizes host cells' vital organs and kill it. They are mostly patristic in immature stage but free-living in adult stage. Parasitoids are mostly small flies or wasps. Few parasitoids attack one or two species and called specialists, while other attack large number of species, therefore are generalists. On the basis of mode of development, OF parasitoids can be categorized as endoparasitoid, internal, or ectoparasitoid, external. The adults are typically free-living, and may be predators. They may also feed on other resources, such as honeydew, plant nectar or pollen. Because parasitoids must be adapted to the life cycle, physiology and defenses of their hosts, they are limited in their host range, and many are highly specialized. Thus, accurate identification of the host and parasitoid species is critically important in using parasitoids for biological control. They includes mostly wasps or flies. They have narrow host range. Commercially, there are two types of rearing systems for parasitoids. Short-term daily output with high production of parasitoids per day. These facilities are used seasonally. Long-term, low daily output systems. These are used on year-long basis. Examples. Parasitoids host. The ichneummonid wasps caterpillars. The braconid wasps caterpillars, aphids. The chalcidoid wasps parasitize eggs and larvae of many insect species, tachnid flies, caterpillars, beetle adults and larvae, and true bugs in Carcia formosa wasp. Greenhouse horticulture. Whiteflies, sap-feeding insects. First biological control agents developed. Gonodocerus ashmedi glassy-winged sharpshooter eastern spruce budworm insect in fir and spruce forests. Life cycles of greenhouse whitefly and parasitoid wasp in Carcia formosa. Encarcia formosa cause wilting and black sooty molds in glasshouse vegetable and ornamental crops. 14.5 Predators. Predator is an organism that lives by feeding on another species. Insect and other arthropod predators are more often used in biological control because they feed on a smaller range of prey species, and because arthropod predators, with their shorter life cycles, may fluctuate in population density in response to changes in the density of their prey. Important insect predators include lady beetles, ground beetles, rove beetles, flower bugs and other predatory true bugs, lacewings, and hoverflies. Spiders and some families of mites are also predators of insects, pest species of mites, and other arthropods. Most of the pests of crops are insects. Mostly insectivorous species are used for biological control. Most of the predators attack wide range of pest species and control their population densities. Examples. 1. Lady beetles. They are predators of aphids. They also consume mites, scales, insects, parasite mealybugs, and small caterpillars. Spotted lady beetle Caleongilla maculata feeds on the eggs and larvae of the Colorado potato beetle Leptinotarsa disseminata. 2. Running crab spider Phylodromus cespetumni are also predators of aphids, leafhoppers, and persilla. 3. Polistes wasp feeds on bollworms and caterpillars of cotton plants. 323. 4. Entomopathogenic nematodes are predators of insects and invertebrates. They generally spread in the soil and infect the insect's host by entering in their hemolymph and releases their bacterial symbionts. These symbionts reproduce and release toxins and kill the host insect. Entomopathogenic nematodes have limited resistance to high temperature and dry conditions. Their effectiveness also depends upon kind of soil. 5. A microscopic nematode Phasmarhabditis hermaphrodita kills slugs. Nematodes get associated with pathogenic bacteria like Moraxella osloensis. Nematode enters the slug through their posterior mantle, but slug is killed by bacteria associated with nematode. 6. The predatory mites Phytocellus persimilis, Neoscylus californicus, and Amblysius cucumaris, the predatory midge Feltiella acarasuga, and a ladybird Stithoris punctilum used for control of spider mites. 7. The bug Aureus insidiosus is used against the two-spotted spider mite and the western flower thrips, Franklinella occidentalis. 8. Aedes aegypti, the dengue-causing mosquito, was controlled by community action of copepods, baby turtles and juvenile tilapia in growing water tanks. Some more examples of predators of crops are predators pests. Big-eyed bugs, lygus bugs, aphids, leafhoppers, spider mites, amoebae, soil-borne fungi, bacteria. Predatory mites, plant-feeding mites. Anthochorid bugs, spider mites, thrips, aphids, parasilla, young scale, various insect eggs, myrid bugs, spider mites, aphids, leafhoppers, parasilla, scale insects. 324. 325. Some of the examples of biological control agents of insects, pests on crops. Vegetables, insect pest biological control agents. Predators, parasitoids, microbial agents. Bendy shoot and fruit borer. Chrysoperlicarnia, Geochorus spp. Eocanthocana. Phyrsellata, coccinellid beetles. Trichogramma chelonis, egg Tetrasticus spp. Egg Telenomus spp. Egg Chelonis blackburni, egg larval Campylettus chloridae, larval Goniophthalmus halley, larval Bracken spp. Larval Obomyrmus albicans, a nematode whitefly myrid bug, Diciphus hesperus, Geochorus sp, etc. Encarsia sp, Erythmaceras sp, Chrysocharis pentheus. Onion thrips, Alothrips spp. Coccinellids. Galindromus. Occidentalis, lacewings. Chrysopa spp. Chrysoperla spp. Amblysius cucumaris. And Amblysius. Barkeri, A. cucumaris. Mites. Serenisis minis, Aureus spp. And Anthochorus spp. Bovaria bassiana metarizum. Anisoplea fungus, Heterorhabditis indica Steiner Macarpocapsae. Heterorhabditis bacteriophora. Maggot braconid wasp. Apherida pallips. Staphylinid N. Aleocara bilinita. Potato tuber moth, lacewing, red ant, ladybird beetle, spider. Robber fly, dragonfly. Etc. Chelonis blackburni, Capodosoma coleri. Trichogramma spp. Apantels sp. Pristomeris vulnerator. Aphids, ladybird beetle, lacewing, spider, hoverfly et. Lycyclobus sp. Diarrhetiella sp. Aphelinus sp. Aphidius. Colmani, etc. Cabbage head borer, Chrysoperlicarnia, coccinellids, Geochorus. sp. Pentatomid bug. Eocanthocana. Phyrsellata. Trichogramma spp. Egg Bracken galecki, larval, bee habiter, larval, etc. Psilomyces spp. Zoophthora radicans, etc. 14.6 Methods for using biocontrol agents. 14.6.1 Classical biological control, importation. Classical biological control or importation involves introduction of new pests, natural enemies, in new or to establish new permanent population. Many organisms that are not pests in their native habitat become unusually abundant after colonizing new areas where their natural controls are absent. For introduction of new species to new area, lots of prior research information is required about pest species, such as their physiological adaptation, negative effect on native species, effect on biodiversity, etc. Natural enemies should be quarantined to prevent their escape until research confirms that they will have little negative impact in the new country of release. Biological control agents should have colonizing ability to new area to keep pace with changes in habitat within space. Control is greatest if the agent has temporal persistence so that it can maintain its population even in the temporary absence of the target species, and if it is an opportunistic forager, enabling it to rapidly exploit a pest population. Negative impact. If species are introduced before prior research, species become serious pests themselves. After selection of suitable pest species, they are kept under quarantine condition to eliminate from any kind of pathogens or natural enemy population. Natural pests are released in field after carefully examining pest as well as natural enemies life cycle for maximum efficiency. Natural enemies are released at proper timing when the target pests are in abundant and disturbance of the newly released enemies is minimized. This is long and tedious process, but results are permanent and effective. It provides long-term benefits over a large area. Examples. 1. The alfalfa weevil Hyperopostica. It is native of Europe and became serious pest in US by 1970s. It is controlled by introduction of natural enemies. Several parasitoids introduced against the pest, out of which 2 were more effective. One species of parasitoid attacked the larva of weevil and other adult. By introduction, weevil population was decreased up to 75% in small time span. 326. 2. Rhodelia cardinalis Vedalia beetle for control of Iceria purchasii, cottony cushion scale. The Vedalia beetle, a predator insect, was imported from Australia to California for control of cottony cushion scale. 3. Control Ophantonina graminis in Texas by Neodusmetia sangwani, the beetle in a parasitoidal fly, Cryptochetum Iceriae. 4. Levuana iridescens, the Levuana moth's population was controlled by biological control program. 5. Trichogramma austrinii parasitoidal wasps used as biological control agent against European corn borer, Austrinia nubilalis. 6. Alternanthera alligator weed, was biologically controlled, introducing alligator weed flea beetle in USA. 7. The giant Salvinia molesta, was controlled by Salvinia stem borer moth, Samea multiplicalis. 14.6.2 Augmentation and inoculation techniques. Augmentation involves the supplemental release of natural enemies that occur in a specific area boosts the naturally occurring populations there. Sometimes resident natural enemies are present as lesser number in their original population, so population is supplemented, by purchasing and releasing commercially available beneficial species for biological pest control. Augmentation is used when enemy populations are lacking or unable to react to the pest population. As a result, unlike importation or conservation methods, augmentation usually does not provide permanent pest suppression. It is direct manipulation of natural enemies to increase their effectiveness. Augmentation requires lots of research work on natural enemies, advanced planning, expert biological advice, regular monitoring, optima, release time, etc., so that certain levels of pests and damage can be tolerated. In inoculation, small numbers of natural enemies are released but at intervals to allow them to reproduce for long-term control to keep the pest population to low level. In contrast, in inundative release, large numbers of natural enemies are released to reduce damaging pests. 327. Population. Inundative releases involve the release of a large number of a natural enemy to the point where their population completely outnumbers that of the pest. Augmentation can be achieved by any of the two methods. 1. Mass production and periodic colonization. It is most commonly used approach in which natural enemies are controlled either inoculatively or inundatively. If in certain locations pests cannot overwinter, natural enemies are released in each spring to population to establish. This approach is more commercially used than others. 2. Genetic enhancement of natural enemies. Other ways to release genetically modified natural enemies. With the help of conventional selection techniques, it has been possible to genetically improve a number of predators and parasitoids. Limitations. It is an effective method, but its guarantee to work is sometimes low. It depends upon interaction between pest and control agent. In some cases, it does not provide adequate pest control. Many natural enemies feed on both pests and beneficial species and reduce the effectiveness of biological control. Examples. 1. The parasitoid wasp, Encarsia formosa, used to suppress populations of the greenhouse whitefly, Triolarodes vaporariorum. Release of low densities inoculation of E. formosa immediately after the first whiteflies are detected on a greenhouse crop can effectively prevent populations from developing to damaging levels. 2. The predatory mite Phytoceolus persimilis is used for control of the two-spotted spider mite. 3. The egg parasite Trichogramma released inundatively to control harmful moths. In response to the presence of pest eggs, these tiny endoparasitoids of insect eggs are really asked in vast numbers in crops or forests. 328. 4. Bacillus thuringiensis and other microbial insecticides are also used. 5. The European native, Trichogramma brassicae, is used to suppress the European corn borer, Ostrina nubilalis, in cornfields. 6. Trichogramma spp. That are inundatively released to suppress sugarcane borer. 7. Nematodes that kill insects are released to soil-dwelling insect pests. 8. Hippodaemia convergens, the convergent lady beetle, is used for biological control of aphids. 9. Beneficial nematodes are inundatively released. They infect insect host by traveling through the soil. Inside insect they release symbiotic bacteria which kill the insect. 10. Laboratory selected, the predatory mite, Metaceolus occidentalis, were commonly used in an integrated mite management. 11. Pediobius foveolatus, isinoculative release in the field against Mexican bean beetles, the parasitic wasp, Edobum putilleri, isinoculative release against the Colorado potato beetle. 14.6.3 Conservation biological control. This is the third method of biological control. Conservation of natural enemies that are already adapted to target pest is simpler and cost-effective method. In order to boost the effectiveness of the beneficial species, it is necessary to discover the factors that may be limiting a specific natural enemy's effectiveness and adjust them. In this approach, natural enemies are preserved by cultural, mechanical, or selective chemical controls in a way that they do not harm the beneficial species. Most of the pests are attacked by multiple species, so conservation can be main way of biological control for them. Conservation of natural enemies involves either providing resources that natural enemies require in their surrounding or eliminating factors that interfere with them. Main conservation strategies include pesticide management. Use of broad-spectrum pesticides sometimes leads to outbreak of secondary pests, therefore it is necessary to reduce the use of broad-spectrum pesticides. Most of hazardous broad-spectrum pesticides include carbamates, organophosphates, and pyrethroids that kill the natural enemies. Even low level of pesticides residues interferes with 329. Reproduction and their ability to locate and kill pests. Neonicotinoids translocate into natural enemies and honeybees that feed on nectar and pollen. Example, spider mite population is affected by application of carbamate and organophosphate. To prevent the long-term control of the pest, non-persistent pesticides provides less harm to natural enemies. These include azadirachtin, insecticidal soap, narrow-range oil, horticultural oil, neem oil, and pyrethrins. Ant control and honeydew producers. This is typically done by soil cultivation around ant nests, encircling trunks with ant barriers of sticky material, use of insecticide baits near plants. If the population of ants is controlled, natural enemies will increase in population and ultimately pest population can be reduced. Example, many species of ants are considered as pests because they consume the honeydew produced by insects, aphids, mealybugs, soft scales, psyllids, and whiteflies. Producers of honeydew are shielded by ants from parasites and predators that could otherwise control them. Habitat manipulation. Various ways of habitat manipulation includes many natural enemies perform poorly if nectar-producing plants are not present to supplement their diet. Planting different species of flower at different times can give regular supply of nectar, pollen, and shelter to natural enemies. Growing diverse species of plants well adapted to local species can retain predators and parasites relationship. These plants can tolerate low populations of plant-feeding insects and mites. Reduce dust. Dust can interfere with natural enemies and may cause outbreaks of pests such as spider mites. Avoid excess fertilization and irrigation. It can cause phloem-feeding pests, such as aphids, to reproduce more rapidly than natural enemies can provide control. Factors interfere with the effectiveness of a natural enemy are: the use of pesticides may result in the direct death of natural enemies or indirect impacts due to a decrease in the quantity or accessibility of hosts. 330. A number of cultural techniques, like tillage or burning crop detritus, can destroy natural enemies or render the ecosystem unsuitable for crops. Repeated tillage in orchards can result in dust deposits on the leaves, which can kill small predators and parasites and increase the population of some insect and mite pests. Examples. 1. In rice fields, nectar-producing crop plants are grown. These plants nectar for parasitoids and predators of plant hopper pests and found to decrease the pest densities by 10 to 100 folds. 2. Aphids predators are found in tussock grasses by field boundary hedges, but their growth is slow. Strip of tussock grasses is planted in the centers of field, enabling aphid predators to overwinter there. 3. Prune trees are planted in grape vineyards to provide an improved overwintering habitat or refuge for a key grape pest parasitoid. 4. Artificial shelters in form of caskets, boxes or flower pots in gardens to attract natural enemies. Example. Earwigs are controlled by hanging flower pots filled with straw or wood wool. Birdhouses are attractive for nesting of insectivorous insects. 5. Periodic washing of citrus tree foliage resulted in increased biological control of California red scale, Aonidiella aurantii. Limitations. Some host plant effects are harmful to natural enemies but to which the pests are adapted reduce the effectiveness of biological control. Some pests have the ability to sequester poisonous elements from their host plant and use them to defend themselves against other pests. The natural enemy's capacity to locate and attack hosts may be hampered by physical traits of the host plant, such as leaf hairiness. 14.6.4 Microbial control, virus, bacteria and fungi. Insects are biocontrolled via various predators, pathogenic microorganisms such as bacteria, fungi, and viruses. They are mostly host-specific in killing natural enemies. Many insects are infected by bacteria, fungi, protozoans and viruses that cause disease. Due to these diseases, growth of insect pests and reproduction slows down. Some bacteria have symbiotic association with nematodes that kill insects by infecting them. 331. Bacteria. Bacteria have limited use in biological control of insects. They can be mainly used for control of other bacteria, fungi, aphids, viruses and scale insects. Bacteria infect insects' digestive tracts. Bacteria are used to control growth of microorganisms in food, water and soil. Example. 1. Bacteria from genera Bacillus is entomopathogenic and mostly used in biocontrol of insects. Bacillus thuringiensis, a soil-dwelling bacterium, is used to control lepidopteran moth, butterfly coleopteran beetle, and dipteran true fly insect pests. These bacteria kill the target insects by use of enterotoxins. These bacteria are available to organic farmers in sachets of dried spores. After mixing with water, they are sprayed on plants like brassicas and fruit trees. Transgenic plants are also prepared using B. thuringiensis. B. thuringiensis var. israelensis used for control of mosquito larvae and the blackfly Simulium damnosum. Vectivac, BTI is effective against C. pipens than M. domestica. 2. Pinabacillus populii bacterium has been used to of Japanese beetle. 3. The bacterial pathogen Paenibacillus glabradula causes high mortality in snails. 4. Bacterium Streptomyces abramideles produces toxins called ivermectins, effective against insect, arachnida and nematode. Fungi. Fungi are mainly used as biological control agents as they cause diseases in insects. Mainly they attack aphids. Fungi can be broadly classified into two categories based on their mode of action: entomopathogenic fungi and nematopathogenic fungi. Examples. 1. The fungus Entomophaga maimega, a pathogen of the gypsy moth. Spores of fungus germinate when gypsy moth larvae are present. Infected caterpillars carry spores for the next winter. 2. Beauveria bassiana is used to control insect pests such as whiteflies, thrips, aphids. 332. And weevils. 3. Lecanicilium can be deployed against whiteflies, thrips and aphids. 4. Metarisum are used against pests including beetles, locusts and other grasshoppers, hemiptera, and spider mites. 5. Polycoelomyces fumoceroseus is effective against whiteflies, thrips and aphids. 6. Purpureosilium lilicinus is used against root knot nematodes. 7. Trichoderma species against certain plant pathogens. 8. Trichoderma viride has been used against Dutch elm disease. 9. Synchytrium solstitial is being considered as a control agent of the yellow star thistle, Centaurea solstitialis. 10. Metarisum and Beauveria are commonly used entomopathogenic fungi used against arthropods. Entomopathogenic fungi. These kill arthropods like insects, ticks or mites. These fungi produce spores as the insect comes in contact with them. These spores germinate in the presence of humidity and penetrate the cuticle of insect, leads to death of insect after 4 to 10 days. Some fungi produce myotoxins that kill insects. Ascomyces with metosporic fungi used for biocontrol of insects. They provide long-term control against larvae and puparia as they recycle them in the soil. Examples: Metarrhizum and Bovaria are used to control mosquitoes. Fungal spores are used as odor traps on indoor house surfaces to control mosquitoes. Fusarium is able to increase dead spikes of obligate holoparasitic weed, broomrape. Legenidium giganteum is pathogenic to larvae of certain mosquitoes. Nematopathogenic fungi. Those fungi that kill nematodes are called nematopathogenic fungi. These includes nematode-trapping fungi. Most of fungi belong to this group. They grow hyphal elements with the host that kills the host. Example: Arthrobatries candida. They have ability to reduce helminths eggs. Drechmeria coniospora. It is effective in destroying larval stages in parasitic nematodes. 333. Endoparasitic fungi. They infect nematode by spores. Example: Harposporium. Anguillulae. Fungal parasite. They invade the eggs of females by vegetative hypae. Example: Verticillium chlamydosporum. Viruses. Viruses are useful in biological pest control. They are used to control the growth of other viruses and microorganisms in food, water and soil. Examples: the Lymantria dispar multicapsid nuclear polyhedrosis virus used against spongy moth. Its larvae are killed by the viruses. These disintegrating cadavers infect other larvae. RNA mycoviruses are used to control fungal pathogens. Baculovirus are widely used biocontrol agents. More than 16 biopesticides are prepared from this group or are under development. CPGV, Cydia pomonella granulovirus, is biopesticide against codling moth, which is pest of apples. Leafhopper infecting virus, HOCV1, Homolodisca coagulate virus -1, kills leafhopper mortality. 14.7 Behavioral control. Most of the insects are considered as pests to crops. They can be controlled by toxic pesticides or by manipulating behavior of the pests. Behavioral control refers to pest management techniques that can manipulate and regulate pest insect populations by taking use of their natural instincts. In behavioral control, large amount of sex pheromones are dispensed within the crop. In this, normal behavior of male is disturbed so that they cannot find the female. Ultimately, mating in different species of insects is interfered. 14.7.1 Types of pheromones. 334. Pheromones are substances secreted by insects and other animals for communication purpose. They affect the behavior of the insects. They can be spread through air, soil, vegetables, or various items. It causes specific types of specific reaction in receiving insects. These reactions include alarm signals, mating signals, mates, aggregation near food resources, warm other predators, food finding, oviposition site protection, sexual determination, etc. Pheromones are target specific and required in low quantity. They also travel slowly but effective over longer distance. Examples: pheromones have important role in insects mating. Some moth travel up to 30 miles to female following pheromone trail. Beet armyworms are a serious pest in cotton. Flooding with sex-attractant pheromones in cotton field, males was unable to find females for more than 100 days. Queen of honeybee secrete a glandular substance, a pheromone passed to all the workers of the colony for coordination of all the activities of worker bees. Benefits: well suited for integrated pest management (IPM) and insecticide resistance management programs. Ideal for use in organic fruit production. Species-specific action works on the pests you need to control. No significant acute toxicity to mammals. Safe to use around beneficial insects and non-target species. Developing resistance is unlikely as it is natural to the insect's mating process. Pheromones can be subdivided into following categories. 1. Sex pheromones. These are types of pheromones produced for mate location and courtship. These are produced by females to attract male for mating purpose. It triggers number of behavior pattern in other sex for mating. However, in some species they are produced by males to attract females. They are very common in insect, particularly in Lepidopteras, butterflies and moths. These pheromones are widely studied till date. In Lepidoptera, sex pheromones are secreted by eversible glands on the terminal end of the female insect and released by sensory receptors of the male antennae, e.g. moths. In 335. Coloptera beetles, they vary in chemical structure. In Diptera, flies and mosquitoes, these are produced by glands opening on the terminal segment of the female abdomen. They are further in males by specialized chemosensory cincilla of the Neal antenna, e.g. Bombyx mori. 2. Aggregation pheromones. These are kind of hormones produced by both the sexes. It brings both the sexes to come together for feeding and reproduction. If these are released in high concentration, they may act as repellent, spacing phenomena. They can overcome host resistance by attack. Other function includes defense against predators. This is beneficial for mating, predation and utilization of resources in both the sexes. They are mainly produced by members of Coleopterus, Columbolan, Diptera, hemisphere species. They are used against pests such as the boll weevil, Anthonymus grandis, the pea and bean weevil, Cetona lineatus. 3. Alarm pheromones. These are pheromones produced to repel and disperse other insects in their area. This causes alarm behavior by which insects are dispersed when some predator arrives. They are mainly found in social or gregarious insects. They are more common in Aphididae and Thripidae. Honey bees, honey wasps, ants, termites, Vespula squamosa produce it when attacked by predators. To prevent incoming predators, Polistes exclamans release alarm pheromones. Their pheromones are elaborated by the mandibular glands, anal glands, or the sting apparatus. They typically lead to flight or aggression in insects. 2-methylheptanone is alarm pheromone secreted by Dolichoderine ants with fruity odor. 4. Epideictic pheromones. These are used to mark territories in insects. In these, insects lays eggs so that other insect would not enter their area. Females of some insects release the epidictic pheromones near their eggs in fruits to signal other females in vicinity to lay eggs at other places. 5. Allochemical. These are substances secreted by insects for reasons other than food. 6. Host marking pheromones. These pheromones reduce the competition among insects of same species. 7. Trail pheromones. Social insects release these hormones for food trails and nests. They have low persistence and are secreted in foraging ants and termites. Ant Formica species uses formic acid as trail marker. Trail marking pheromones Isoptera, termites, 336. Consists of various substances. Examples: hexanoic acid in Zeudermopsis sp.eg, leaf-cutting ants, Attataxana. Some ants make their path with hydrocarbons as signal to return home. These trails serve as a guide for other ants. 8. Apneumone. It is a pheromone released by a non-living substance that causes a behavioral or physiological response that is adaptively beneficial to the receiving organism. 9. Cinnamone. A material created or acquired by an organism that, when it comes into touch with a member of another species in the wild, causes the recipient to respond behaviorally or physiologically in a way that is advantageous to both the emitter and the receiver in terms of adaptation. 14.7.2 Uses of pheromones in pest management, monitoring, mass trapping and mating disruption. In crop management, trap cropping, the process by which pests are attacked to protect valuable resources, has been used from long time. Some of the methods of pest management are enlisted below. Monitoring traps. Sex pheromones are widely used to monitor insect pest specificity, selectivity and abundance. Sex pheromones traps are designed to attract individuals, usually males with a sex pheromone. Traps based on pheromones are also used for monitoring. Trapping provides information that is essential in the timing of other control methods. In the case of traps, several attractants can be used like pheromones, food and attractive colors. Pheromone bait traps help detect density and presence of insect pests. Food attractants are used to monitor pests such as fruit flies in horticulture. Food baits are used for attraction of insects and pests. These baits consist of brown sugar, sucrose, hydrolyzed corn protein and wine vinegar. Monitoring by visual stimuli by colored trap is also possible. In this method, insects are exposed to colored surface impregnated with glue. Example: Indian meal moths, pantry moths, gypsy moths, Japanese beetles are attracted to a pheromone in a small box lined with a sticky substance and are thus captured for disposal. 337. Pheromones bait traps. For monitoring, sticky traps are placed in a field or orchard to catch them. Later on, their number is monitored. Numbers of males are counted every week to monitor development of population. Examples: cockroaches and stored grain pests. Monitoring allows spatial and temporal distribution of the insect. In pest monitoring, several devices such as traps can be used aiming to quantify specific insects. Advantages. It is efficient and economical method of pest management. It reduces cost and sampling time. Precautions. Trap baits must be selective in insect capturing. Trap location and its height in the field should be determined properly. Monitoring tools must be efficient enough to capture the target pest at low densities. It must be low cost and easy to carry. Mating disruption. In this, males of insects are confused by filling with similar pheromones in the air, so it hinders their mating process. In presence of excessive amount of pheromones in air, males are unable to locate females. In this method, large amount of synthetic sex hormones are distributed in field to disturb mating. 338. Males are unable to find natural pheromones by females, thus unable to inseminate the females. This method is also called male inhibition technique. Example: control of pink bollworm. Examples: normally, males of moth locate female by sex pheromones secreted by females. But when the location is saturated with appropriate pheromone, male moth gets confused, leads to mating disruption. Pink bollworm PCC rhinophorus gossipielu controlled using gossipler. 339. This method is useful against orchard pests as oriental fruit moths in peaches, codling moths, peach tree borers, lesser peach tree borers, and dogwood borers. Advantages. It is effective method of reducing the population of harmful insects. It also limit the use of insecticides. In some cases, insects are eradicated from their areas. Limitation. Although it eradicate the primary pest, but sometimes lead to outbreak of secondary pests. Mass trapping, attract and kill. This method is extension of mating disruption in which insecticide is used along with pheromones. This is a technique by which large numbers of target insects are removed from the population for IPM, integrated pest management. It is used to control back beetles with aggregation pheromones. Example: effective against alfalfa loopers. Mass trapping is the method to reduce the number of individuals of the next generating by using males of both the sexes of the area. It is done in combination of other method for control of pests. Example: whiteflies can be trapped with this method. 340. Advantages. It is newly appreciated, highly effective, environmentally friendly and reasonably priced method of reducing populations of insect species that are susceptible to this approach due to pheromone communication systems and bionomical characteristics make them. In this, experimental traps are placed in large number to collect large number of insects. Example: American palm weevil, Rhynchophorus palmarum, was collected using pheromone traps. These species damage coconut palms. 14.8 Genetic and biotechnology control. Biotechnology and genetics are tools that has recently been used for manipulating gene of biocontrol, and efforts are been made to enhance their expression in biocontrol agents. Biotechnology uses biological agents or living system for manipulation of living organisms, plants and pests. For these purpose, bacteria, fungi, nematodes and insects are manipulated. For example, insects are genetically modified for advanced production of toxins, scorpion toxins, mite toxins, etc., production of metabolic enzymes to enhance production of killing substrates, virulence and host specificity. They are called as GMOs, genetically modified organisms. They are largely being used in insect pest control in agriculture, safe for human and mostly environment friendly. Example, Bacillus thuringiensis, which codes for insecticidal protein. In pest management, biotechnology approaches broadly categories in two categories. Conventional biotechnology. It involves whole range of bioagents, tissue culture techniques and biofertilizers. Bioagents consist of microorganisms, natural enemies, parasites and botanicals. Bioagents mostly composed of bioagents and their secondary metabolites. To maximize their benefits, these agents are used in combination. Plant tissue culture technology has also emphasized in insect pest management. It involves multiplication of disease-free plants under aseptic conditions. By this technology, secondary metabolites are produced and it is also helpful in disease-resistant plants. Modern biotechnology. These involve non-transgenic or organisms that do not involve gene transfer. With the help biotechnology, biopesticides are also being prepared. Viruses are studied that act specifically on host arthropod species like agriculture pests. Encapsulated biofertilizers are Nano 341. Fertilizers are also prepared by use to genetic manipulation. In recent years, encapsulated biofertilizers or nanofertilizers are also found to be effective. Other important biotechnology agents are bionano agents, parasites, biopesticides, etc. Baculovirus recombinants produce Bt toxins. These biopesticides have efficiency than wild-type baculovirus. Nanotechnology, another field related to biotechnology, is largely being used for insect pest control, plant pathogen and insects. It is highly effective and green technology. Nanoparticles are for formulation of pesticides, insecticides and insect repellents. Molecular breeding is another way in which DNA markers are employed in plant genome to analyze characteristics like genetic variability, fingerprinting, population genetics, plant breeding, etc. These markers include RFLP, RAPD, SSR and SNPs. These markers are applied at seedling stage to manipulate the traits. 14.9 Insect attractants, repellents and antifeedants. Attractants. Attracts are substances used to attract in insect pest management. Attractants are active in minute quantity and changes the behavior of the insects and help in pollution and density regulation of insects. Mostly behavior like search for food, shelter, mating activity, aggregation, oviposition, etch are manipulated. Semiochemicals. Semiochemicals are chemicals that stimulate and use for interactions amongst organisms. These chemicals manipulate the behavioral in insects. They include intraspecific pheromones and interspecific allochemicals. They can be further divided as allomones, chiromones and pheromones. Allomones or interspecific semiochemicals provide advantage to producer species. Chiromones provides adaptive advantage to the receiver species. They are products of secondary metabolites. They are natural chemical substances present in plants that direct the insect pest towards suitable sites of feeding. They function as olfactory stimulants, produce orientation behavior similar to sex pheromones. The third categories include pheromones and are intraspecific semiochemicals. Please see section 14.7.1 for details about pheromones. 342. Repellents. Repellents are chemical substances that make insects move away from food resources. They prevent insect damage to plant by portrait them unattractive and offensive. They include wide range of chemicals. Naturally occurring feeding repellents exist for many insects and are helpful in host selection and specificity. Examples. Dimethyl phthalate and 2-ethyl-3-hexanediol. It is used as repellent against biting flies. And mosquitoes. Bordeaux mixture and tetramethylthiorum disulfide feeding detergent to foliage feeders. Bordeaux mixture consists of copper sulfate, hydrated lime, and water and used as repellent to flea beetles, leafhoppers, and potato psyllid. Tetramethylthiorum disulfide repels Japanese beetle, Popilliu japonica. 4-D-methyltriazono acetanilide, which is a feeding repellent used against foliage feeding insects like the cabbage looper, the cotton leaf worm, cotton boll weevil, spotted cucumber beetle, etc. 343. Natural repellent. 6-methoxybenzoxazolinone, 6-MBOA. It provides protection to European corn borer creosole or 4,6-dinitro-O-cresol. They are soil barrier against bug migration from small grain to corn wood feeding insects. Some species of wood are resistant from termite attack. Example, penicillin monomethyl ether in pine, taxophilin in Douglas fir and p-methylanthraquinone from the Indian teak. Treating with chlorpyrifos EC also prevent termite attack. Fabric-eating insects. Colorless dye stuffs Ulins and Metine FF can be firmly affixed to wool during the dyeing operation and provide protection over lifetime. Sodium aluminonyl fluosilicate is effective repellent for fabric pests. Antifeedants. Antifeedants are substances that inhibit the feeding of the insects, kill them by making them starve. It inhibits feeding of insect on plant rather than keeping them away, unlike repellents. They work by acting on taste receptor of the insects and inhibit their perception of the stimuli. Some of the examples of antifeedants are triazines, herbicides, organodons, fungicides, carbamates, insecticides, natural antifeedant, neem, Indian lilac, Azudiruptu indicu. 14.10 Summary. Many technologies and strategies are being used for control of natural enemies, including large use of chemicals. But chemical methods result in rising resistance and have environmental risks. Biological control is suitable alternative to chemical control. It includes use of predators, parasites and pathogens. Biological control is especially effective in agriculture sector to control parasites. However, sometimes biocontrol agents have negative impact on biodiversity. With recent advance in genetics and biotechnology, biocontrol can be used in more effective ways. In this method, new biological control agents are identified and older ones are genetically engineered to 344. Make them more effective against natural enemies. Thus, in future, this strategy will become more effective than chemical control method. Sericulture is the process of breeding and managing silkworms for commercial silk production. Silkworms are raised on various mulberry plant species. The silkworm is the caterpillar or larva of the domesticated silk moth, Bombyx mori. Silkworm rearing entails a series of events such as the collection and processing of silkworm cocoons in order to extract raw silk fibers. Raw silk is yarn made from cocoons spun by certain insect species. The main activity of sericulture is the collection of silk filaments, which provides millions of people with high-paying jobs with little capital investment. 300. (347) Mulberry and non-mulberry sericulture. There are 5 major commercially important types of silk. Mulberry types are those that are grown on mulberries, while non-mulberry types are those that are not grown on mulberries. Non-mulberry varieties are obtained from various species of silkworms, which feed on a variety of food plants. Other than mulberry silks, other types of silks are known as non-mulberry silks. Types of silkworms. Mulberry type. Non-mulberry types. Tassar. Oak tassar. Airy muga. Mulberry type. Silkworm, Bombyx mori. Mulberry silk is produced from the silkworm Bombyx mori, which only consumes the leaves of the mulberry plant. The majority of silk that is sold commercially is obtained from mulberry silk. Silkworms for mulberry silk are completely domesticated and reared indoors. Major mulberry silk producing states in India: Karnataka, Andhra Pradesh, West Bengal, Tamil Nadu and Jammu and Kashmir. Non-mulberry silk types. Tassar silk. Silkworm, Anthorea mylitta. Anthorea mylitta, which feeds on the food plants asan and arjun, is the primary source. It is raised in the open trees under natural conditions. Its culture is primarily carried out by tribal communities in India. This copper-colored silk is used for furnishings and interiors. It lacks the luster of mulberry silk but has its own distinct feel and appeal. 348. Major mulberry silk producing states in India: Jharkhand, Chhattisgarh, Orissa, Maharashtra, West Bengal and Andhra Pradesh. Oak tassar silk. Silkworm, Anthorea proili. It is fine silk obtained from Anthorea proili, which feeds on oak plants found in abundance in India's sub-Himalayan belt. Major mulberry silk producing states in India: Manipur, Himachal Pradesh, Uttar Pradesh, Assam, Meghalaya and Jammu and Kashmir. China is the major producer of oak tassar in the world. C. Airy, Endi or Arundi silk. Silkworm, Phyllosamia ricini. It is made of multivoltine silk spun from open-ended cocoons. Its silkworm is easily domesticable. Phyllosamia ricini is a parasite that feeds on castor leaves. It is also grown by tribal communities because the pupae are high in protein. Silk is used by tribals to make chatters, wraps. Major mulberry silk producing states in India: the northeastern states, Assam, Bihar, West Bengal and Orissa. Muga silk. Silkworm, Anthorea assamensis. It is golden yellow silk produced by Anthorea assamensis, a multivoltine silkworm that feeds on the aromatic leaves of sam and solu plants. It is semi-domesticated varieties. Muga silk is used to make saris, mekhalas, chatters, and other items. Major mulberry silk producing states in India: Assam. 15.3 Cultivation of food plants. 15.3.1 Mulberry plant. 15.3.2 Mulberry species and varieties. 15.3.3 Soil and climatic conditions. 15.3.4 Establishment of mulberry garden in first year. 349. 15.3.5 Maintenance of mulberry garden. 15.3.5.1 Pruning. 15.3.5.2 Biofertilizers and micronutrients. 15.3.5.3 Intercropping. 15.3.5.4 Weed management. 15.3.5.5 Harvesting. 15.3.5.6 Leaf yield. Basic steps of cultivation of mulberry plants are: initially, land is selected and prepared after plowing, leveling, manuring and so on. After sufficient rainfall, saplings are planted. The distance between 2 samplings should be 20 and 25 feet. Watering, manuring, and plowing of the soil around the samplings are done at predetermined intervals. Bushes are pruned every 3 to 5 weeks before the start of rearing season. Because the plant will not have enough foliage to rear worms every year, the land has been divided into 2 plots. It is necessary to provide proper care and attention to host plants at regular intervals in order for offshoots to grow quickly and healthily. This includes plowing, manuring, watering, and pruning. A. Mulberry plant. Host plant of mulberry silkworm. Silkworm feed on mulberry tree leaves, which are highly digestible of them. The leaves and young stems are high in protein and minerals. The leaves and young stems are high in protein and minerals. There are no anti-nutritional or toxic compounds in it. Variety, location, average temperature, solar radiation, rainfall, plant density, fertilizer application, and harvesting techniques all influence plant yield. Mulberry silkworm is a monophagous insect that only feeds on mulberry leaves. More found in the leaves attracts the silkworm. B. Mulberry species and varieties. Classification of mulberry. Division phylum Phanerogams, flowering plants. 350. Subdivision subphylum Angiosperms. Class Dicots. Order Urticales. Family Moraceae. Genus Morus. Species Morus alba. Species and improved cultivars of mulberry. Important species: Morus alba, M. lobogata, M. bombycis, M. indica, Morus nigra, M. serrata, M. lavigata. Mulberry varieties. Irrigated, Canva 2, Mr. 2, S30, S36, S54, DD Biswa V1 semi-irrigated, Canva 2, Mr. 2. Rainfed, S13, S34, RFS 135, RFS 175, S1635. Distribution: Morus alba, M. indica, M. serrata and M. lavigata grow wild in the Himalayas. For the purpose of rising silkworm, M. indica and M. alba are grown in the hilly and plain regions of India, Himalayan region. Maharashtra and Meghalaya are where you can find M. indica. Northeast and central India are home to M. lavigata. The red mulberry, Morus rubra, is a native of North America, and the black mulberry, Morus nigra, naturally grows in Western Asia and the Middle East. Tassar silkworm. Host plants: Argent Terminalia arjuna, Asan, Terminalia tomentosa, Sal, Sharia robusta, Patuca, Melastoma melabathrium, Bogori, Ziziphus jujuba, Antharea proili, temperate species. Muga silkworm. Host plants: Antharea asama, Anibeti, Acampta and Ahelferi, Mochilus bombicina and Solu, Litzia monopetela, Digloti, Litzia salicifolia, Mahankari, Litzia cubiba, Bogori or beer, Ziziphus jujuba, Champa, Michelia champaca, Bomloti, Simplocus 351. Grandifolia patihunda, Actinodaphne obovata, Gamari, Gamelina arborea, Panchapa, Magnolia sphenocarpa, Cathalua, Cyclocodaphne natita, Ganseri, Cinnamomum blandoliferum, Bodramoni, Xanthoxylum resta. Airy silkworm. Host plants: Airy, Samia ricini and Samia cynthia, Atticus atlas. Cricula species: Castor, Ricinus communis, is primary food plants of airy silkworm. Several alternative food plants: Arborkeseru, Ailanthus excels, Barpat, Ailanthus grandis, Topioca, Manahat esculenta, Gulancha, Plumeria acudifolia, Gamari, G. melina arborea, Payum, Avodia flaxinifolia. C. Soil and climatic conditions. Mulberries can be grown up to 800 meters. Optimum temperature range for growth of mulberry: 13 degrees Celsius to 37.7 degrees Celsius is the temperature for mulberry growth. The ideal temperature is from 24 and 28 degrees Celsius with a relative humidity of 65 to 80%. Sunshine, of 5 to 12 h per day. Rainfall, 600 millimeters to 2,500 millimeters per year. Soil conditions: plants grow in loamy to clayed loam soils. It can withstand acidic soil conditions. In the case of alkaline soils, pH below 5 can be maintained by applying dolomite or lime. Gypsum is sometimes added as well. Mulberry grows best in soil with a water retention capacity of 40–45%. Mulberry plants grow best in slightly acidic soils, 6.2 to 6.8 picohenries, that are free of harmful salts. Soils that are saline or alkaline are not recommended. De-establishment of mulberry garden in first year. A nursery land preparation. In general, 800 sqm area of red loamy soil is selected in the main field, to which manure is added and proper drainage is provided. 352. B. Nursery bed preparation. It is mostly done before the arrival of the monsoon because there are a lot of seedlings available before the monsoon. Nursery can also be prepared from mature seed after the monsoon. Seeds can be directly used to raise plantations and transplant them in the field to shorten the establishment period. C. Bed layout, bed size. Choose well-drained high land in a shady location. The soil is dug or plowed twice or 3 times up to a depth of 30 centimeters, then properly leveled. Seed beds of a suitable size have been prepared. D. Pre-treatment of cuttings. In 40 liters of water, 1 kilogram of azospirillum culture is added for cutting treatment. Before planting, soak the bottom end of the cutting for 30 minutes. Azospirillum is used to promote early rooting. E. Planting and management of nurseries. The nursery must be irrigated every 3 days. By adding 1 kilogram of malathion 5D and QUINALPHOS 1.5D, termite infection can be avoided. The soil is treated with carbendazim 50WP or trichoderma viride to prevent root rot and collar rot. Urea is added after weeding. F. Sapling age. After 90 to 120 days of planting, the saplings are ready for transplanting in the main field. Methods of irrigation. It is artificial application of water to soil in order to access the crop production. It supplements the water available from rainfall and groundwater. Ridges and furrows method, flatbed method, drip irrigation. 353. 354. Irrigation requires less water. During heavy rains, furrows act as drainage channels. Water infiltrated the soil and spread throughout the root zone. It lowers evaporation losses. A low-cost method. In clay or deep clay soils, a shadow furrow is formed along with a guiding ridge to prevent soil cracking. This type of furrow is known as a corrugated furrow. Rectangular beds and channels form. Water runoff is relatively low. More land is wasted. More labor is required for field preparation. Reduced water loss. Improved crop growth. Suitable for undulating terrains. Ability to apply fertilizers in addition to irrigation water. Clogging of emitters by physical, chemical and biological pollutants. High initial cost. Propagation of mulberry. Mulberry can be propagated in two ways. Sexual, asexual. Sexual propagation. It is done through seedlings. For this, seed population is selected and hybridization is done. Seed selected for propagation should have following qualities. High quality seed. High germination rate. Seed must be collected from fully ripened fruits. Seed must not be damaged. Merits. Sexual propagation introduces variability in the progeny and gives scope for selection of new varieties. Demerits. Comparatively long gestation period to provide leaves for silkworm rearing. The desirable traits of improved cultivars cannot be perpetuated. Raising seedlings. Seedlings can be raised by sowing seed in soil directly or in polytubes. Pre-treatment of seeds and seed dressing. Before sowing, seeds are soaked in water for 20 to 24 and treated with babaston or carbendazim. Seed sowing. Seeds are sown in lines in the prepared beds at a spacing of 15 centimeters in the row and 15 centimeters between rows at a depth of about 2 to 4 cm. Seed germination. After 30 to 45 days of seedling, seed germination begins. However, polyembryonate seedlings are also known to exist. Normally, one seed only produces one seedling. Twins and triplets have been seen in seedlings from polyembryonate embryos. Transplantation of seedlings to polytubes. 1. Polythene tubes can also be used to grow the seedlings. For nursery preparation, seedling of 6 months old and 10 to 20 centimeters tall is gathered. After being collected, the seeds are placed on a moist bed shaded by a tree, covered with moist gunny bags, and sprinkled with water to keep them moist. 4 weeks later, the germination process begins. Daily sorting and individual sowing of the germination seeds takes place in 9x 6 inch, 150 to 220 gauge polythene tubes that are filled with rooting material and maintained shaded by trees. 2. The rooting media for growing seedlings in polythene tubes is a mixture of soil, sand, and farmyard manure (FYM) in the proportions of 1:1:1. Asexual propagation. Vegetative plant parts are used in this type of propagation. The parent stock's inherited features can be preserved through asexual reproduction. The desired characteristics can be carried to next generation. 3 basic types of propagation are used in mulberry. 1. Cutting. 2. Grafting. 3. Budding. The most common method of mulberry propagation is cutting. Plants can either be planted directly in the ground or they can be started in a nursery and then transplanted into the main field as sprouted and rooted saplings. 355. Precautions while selecting proper plant for cutting. The cutting material should have a diameter of 10 to 12 mm and be free from insect and diseases. The mulberry plants are grown from cuttings of semi-hardwood. Cuttings are selected from well-established, 8 to 12 months old gardens. Cutting material should have 3 to 4 active buds, be between 15 and 20 centimeters thick and have a 45-degree slanting cut at the bottom. Methods of plantation. Row system. By creating ridges and furrow, the land is prepared using this technique. Between two ridges, there is a 45-60 centimeter space. Water used for irrigation runs through the ruts between the rows, and in most cases, bush mulberries are produced and collected via bottom pruning. Pit system uses a greater spacing, is used for crops that are fed by rain. Instead of plowing the entire field, standard-sized pits 40 x 40 x 40 centimeters are dug with 90 x 90 centimeters between plants and row for a bush type of agriculture. After mixing equal amounts of organic manure, red soil, and sand in each pit, a cutting or a sapling is planted. It is initially watered every day until it roots. The trenches are larger 45 x 45 x 45 centimeters if tree-type plants are to be grown on hedges, the side of the road, etc. Maintenance of mulberry garden. Pruning. After a year of planting, mulberry tree are pruned. There are 3 ways to do it. Ground bottom pruning. The plants are clipped at the ground, leaving a stump that is 10 to 15 centimeters above the ground level. Once in a year, this kind of pruning is carried out. This kind of pruning involves cutting of branches at the stems at ground level base. Middle pruning. At 40 to 60 cm above the ground, the branches are cut. Following bottom pruning, subsequent cuts are made at a height of 45 to 50 cm. Cutting bush mulberry at a height of 45 to 60 cm above the ground is known as middle pruning. This approach is used in regions with lower rainfall, flood-prone fields and larger mulberry farms. 356. Koehler or strip system. This method of trimming is done in densely planted areas. Every time, the branches are cut at ground level. As a result, it undergoes 5 prunings per year. It's necessary to fertilize heavily and irrigate after this kind of harsh pruning. At each harvest, the entire stalk is removed and cut to the ground. This is only appropriate in regions where mulberries sprout all year round without a latent season for bud sprouting. Top clipping. This technique entails only cutting off the branch's top. It is typically practiced in the winter. It offers greater benefits for growing and harvesting high-quality mulberry leaves. Since it is merely a certain branch of training, it has no impact. Advantages. Leaf yield can be improved through pruning, and leaf production can be coordinated with silkworm rearing schedules at all times of the year. Beet pruning schedules regulate mulberry branches' irregular growth, preventing wasted. Food and energy. Beet pruning should be done closer together and at a height of 3 to 8 centimeters above the ground. In under-row systems, various pruning techniques include a fist form, refers to the mulberry plant being cut annually at the same location on the main stem. After a few years, the portion thickens and takes on the shape of a fist, therefore, the name. With this approach, mulberry diseases and pests are simple to control. Beet non-fist form. Pruning is carried out in such a way that 2 to 3 branches are permitted to sprout from the main stem, basal sections, and secondary branches also developed as a result. Resistance to disease and pest control is considerably lower using this approach. Fertilizers and mineral nutrients, biofertilizers. Azospirillum. Phosphobacterium. 357. 358. Beet micronutrients. Various micronutrients are used depending on the symptoms of deficiency. Zinc sulfate, ferrous sulfate, borax, copper sulfate, manganese, or sodium molybdate are a few examples. Intercropping. Mulberry may successfully intercrop with a variety of vegetables. The farmers benefit financially from it. The farmers may receive additional income as a result. Advantages. Short-duration pulse intercropping improves the soil, generate extra income, and inhibit weed growth. Weed management. Cultural method, mechanical method, chemical method. By using this procedure, weeds' stump roots are eliminated. Hand hoes are used to eradicate weeds. In this paraquat, gramixone at 2 to 3 lit per hectare is used. Ammonium sulfate liter is also used. Abutilin indicum velvet leaf Amaranthus viridis pigweed Califa indica copper leaf Borhyavia diffusa hogweed Croton sparsiflorus. The following benefits come from mulching with clipped mulberry twigs as well as other materials like straw and dry leaves. Controls the development of weeds. Preserves soil moisture by decreasing runoff. Increases water infiltration. Lower soil temperature. Harvesting. The technique used to collect leaves is determined by the type of rearing used. The best time to collect the leaves is early in the day. Mulberry leaves can be collected using one of 3 techniques. Leaf picking. Individual leaves, with or without petiole, are harvested. Leaf picking begins 10 weeks following bottom pruning, with subsequent picking spaced 7 to 8 weeks apart. Branch cutting. The entire branches are cut and fed to the worms. To ensure that the lower leaves mature uniformly, topping is done first. Beet whole shoot harvest. Using bottom pruning, the branches are chopped at the ground level. Shoots are harvested every 10 to 12 weeks, resulting in 5 to 6 harvests each year. Time of harvest. It is preferable to harvest the leaves in the morning. Preservation of leaves. To keep the leaves cold and fresh, cover the bamboo basket with moist gunny sacks or store them in leaf preservation chamber. Yield of mulberry leaves. Under a row system of production, an average of 25 to 30 mt of leaves without twigs can be gathered annually per hectare. 15.4 Rearing of silkworm. 359. Basics of silkworm culturing. The caterpillars are maintained in bamboo trays that also contain freshly chopped mulberry leaves. The caterpillars stop eating the leaves after about 30 to 40 days and then moves inside the small chambers in the bamboo trays to spin cocoons. These are produced when their salivary gland secretes the liquid protein. Small racks are given in the trays so that the cocoons can cling to them. The cocoons are where silk moth develops. A silkworm goes through a complete metamorphosis, holometabola. In 45 to 55 days, mulberry silkworm's life cycle completes. The stages include egg, larva, pupa and moth. The egg stage lasts for 9 to 10 days, the larval stage lasts for 24 to 28 days, the pupal stage for 8 to 10 days and the moth stage is 3 to 4 days. Life stages of silkworm Bombyx mori. 1. Egg. A silkworm eggs weighs about 2,000 eggs, or 1 gram. The season affects the egg's size and weight. A multivoltine breed from India produces 400 eggs every laying. The outer covering of egg is called corion. Egg oozing is referred to as micropyle. 2. Larval stage. Newly hatched larvae are 3 mm long and are referred to as antarchigo. The body is coated in bristles, and the head is huge. It has 4 pairs of tubercles, i.e., subdorsal, suprasporacular, intrasporacular and basal tubercle. It takes 20 to 24 days for it to molt. There are 5 phases of larval life. The 5th instar larva is produced after molting. The mulberry leaf is the primary food source for phytophagous, plant-eating larvae. The head, thorax, and abdomen are the 3 sections of the body. The thin elastic chitinous cuticle permits rapid growth of the larva during any instar. Distinct features based on voltinism. Univoltine. The larvae have large bodies and a prolonged growing period, making them suitable for cooler climates. The quality of silk and cocoon is excellent. High temperature and high humidity are inappropriate for growth. Bivoltine. It is suitable for warmer environments. Shorter larval duration than univoltines. The quality of silk, cocoon and larva are all excellent. Moderately suited to temperature over 100 degree and extreme humidity. 360. C. Polyvoltine. Adaptable to warmer climates, shorter larval period, stronger larva. Tiny cocoon, low silk quality. Solely produces non-hibernating eggs. 3. Pupa. It is encased in a cocoon. The cocoon is made up of silk fiber secreted larvae in their 5th instar. Pupa resides within the cocoon. Pupa does not move or eat. Pupa is made up the head, thorax and abdomen. The pupal stage lasts approximately 10 to 12 days. 4. Adult. The adult emerges from pupa. Adult life lasts approximately 3 to 5 days. The adults do not consume food. They exist solely to reproduce. They die after copulation. Varieties of silkworms. 1. Mulberry silkworm Bombyx mori. 2. Tassar silkworm Antheraea mylida.